Structural Biology Core was created in 2002 to allow researchers at the Institute to solve structures of macromolecular complexes so that they could further their research by understanding how these molecules behaved at the atomic level. To carry out this research, we have state-of-the-art crystallization robotics, we have imaging systems, as well as the state-of-the-art in-house X-ray crystallography generator and detector that allow us to do everything from setting up the crystallization trials to solving the structure.
We solve the structure, we analyze the structure, and we look at it and say, okay, based on what you're telling us, this is what we think that the protein is and does and how it works. And we might be able to guide the other scientists into the next set of experiments.
The goal is to learn essentially how the protein functions mechanically and in terms of how it binds different ligands and substrates.
One of the real benefits to having a structural biology core is it allows investigators to get hands-on experience in these techniques if that's something they want to do. On the other hand, if they would prefer just to have the results of these types of experiments, then we can do that research for them.
It's a very organic sort of process because crystallography it's not a guarantee. And so you have to try a lot of different things sometimes to get it to work the way that you want it to. I had one project where I had worked on it for years and years and years and it just wouldn't work. I'm going to try this one last thing. I didn't expect it to work, but it absolutely did. Beautiful crystals that diffracted miraculously well. And we've solved 20 or 30 structures that way, just from one idea that may or may not work. But you got to give it a try. And if it does, it really is the best experience ever.
One thing for certain that they have that's really important is actually up here. It's the brains. They're very easy to get along with and to work with and to communicate with.
You need help or you have questions, you just need to knock at the door. No matter what's the question, they always provide very full assistance and deep expertise.
We can sit down with you and help you design your experiment. If you don't have the resources in your lab to carry out those experiments, you can carry it out in our space, or we can perform these experiments for you. We're capable of doing the molecular biology, the biochemistry, the protein expression, protein purification. We're capable of doing the crystallization, solving the structures, and helping you interpret what those structures mean and how they relate to the questions that you're asking. At the NIEHS, we're equipped with an in-house Rigaku Synergy R rotating anode X-ray generator system. This is equipped with a high-pix detector that allows for direct single-photon counting. This system also has automated slits that allow the user to adjust the X-ray beam so we can optimize for either high brilliance or low divergence. This is the ideal system for collecting in-house publication quality data, or it can be used for a screening tool in order to send crystals to the synchrotron. The Structural Biology Core also helps secure synchrotron time at either Brookhaven or Argonne National Laboratories. This allows us to collect the highest resolution data possible, as well as work on crystals that would be impossible to study on our home source. We have the ability to characterize samples using techniques such as differential scanning fluorimetry, light scattering, and isothermal titration calorimetry. This really allows us to thoroughly characterize our samples in ways that are useful for structural determination and follow-up studies. To help with crystallographic screening, we have mosquito robots that can set up 288 crystallization conditions in just a few minutes. These instruments greatly enhance our throughput while minimizing the amount of sample required. One of the real challenges with setting up so many crystallization screens is going through them all to determine which conditions actually gave positive hits. At 4 degrees, we're equipped with a Rock Imager 182 imaging robot. This instrument is capable of housing 182 96-well trays that it screens on a schedule using both UV and visible light. The core stocks a wide variety of commercial crystallization screening conditions, so the individual NIEHS investigator doesn't need to make a significant investment to purchase their own screens.
We saw the crystal structure of the enzyme and substrate complex with 3 different states that allow us to outline the catalysis mechanism.
Three-dimensional structure determination is very rewarding because you get this three-dimensional model when you're done. It's so much information And it's very satisfying knowing this is something that's actually at the atomic level, and I've developed this model that we can look at.
I will say that things have moved very quickly in a good way because of the technology that they have. The turnaround time is very quick, and that also is a testament to how hard they work.
Sometimes when we try to crystallize proteins, we don't always get something on the very first try. So our lab has developed several different rescue strategies that involve fixed arm chaperone crystallization, surface entropy reduction mutations. We can use nanobodies. We also like to use specific binding partners or ligands in order to help stabilize the proteins and crystallize them in a biologically relevant form. One of the exciting things about working in the structural biology core is working on a wide array of biological problems with many different scientists. Our work has contributed to a better understanding of how DNA polymerases function, how DNA double-strand breaks are repaired, how flame retardants might function as endocrine disruptors, how allergens can be changed to improve immunotherapy, and how enzymes can be altered to improve chemoenzymatic synthesis of oligosaccharides for therapeutics. There's a lot more opportunity for exploration here.
It's really fun to interact with collaborators, to show them the structure of a protein they've been working on for 10, 15 years, which they've never seen before, and they get to see it and they get to understand now how that protein functions or how it interacts with other proteins. It's really exciting to see how we're able to contribute to their research and expedite and further what they're able to do.